Hello, everyone, and welcome to today's webinar, Expanding Proteomic Horizons with Single Molecule Proteome and Proteoform Analysis. I'm Cassie Soltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Nautilus Biotechnology. To learn more, visit www.nautilus.bio. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. Also, if you experience any technical issues, you can utilize this Q&A box and we will assist you directly. I'd like to now welcome our speakers. Parag Mallick, founder and chief scientist for Nautilus Biotechnology, and Andreas Hühmer, senior director for scientific affairs and alliance management at Nautilus Biotechnology. Parag, you may now begin your portion of the presentation.
Thanks so much, everyone, for joining us today. I'm really excited to dive into the different aspects of the Nautilus platform. And I'd like to just start off by recognizing how tremendous a year we've had in proteomics. There have been so many advances across different diseases, breast cancer, Alzheimer's disease, and more. It's astonishing to see how much proteomics is growing and having a tremendous impact. But despite the tremendous progress, if we look at this figure on the right, we can see that it continues to be true that genomics is outpacing proteomics. And really at Nautilus, our goal is to bring proteomics into parity with genomics. So it is a standard part of biological workflows. Today, what we're going to focus on are four things. We'll dive into what the Nautilus platform is, how we think about it, what we're aiming to build, and why. We're then going to dive into some common questions that we've heard from folks about the platform, as well as our most recent advances to share with you on the broad scale side of the platform. And then Andreas is going to take over and talk about some really exciting results we've had in studying proteoforms. To start off, I'd like to really say where we started as a company was taking a step back and thinking about if we were to design an entirely new platform from scratch, what might we want it to do? And as we looked to the community and listened extensively, we heard a couple things. Number one, we want a platform that's comprehensive, that's able to measure substantively everything that's in a sample. We also want a platform that's extremely sensitive so that we can potentially measure those extremely rare, extremely important proteins. So for instance, transcription factors in cells. We of course need a wide dynamic range to match the scale of the proteome. It is an analytical platform, so it needs to be reproducible and robust. We also have heard loud and clear that runtime is critical. People want to crunch through a large number of samples, and we want to be able to enable that. And then critically, we ultimately want a platform that is able to be democratized. So it's accessible to anybody, something that's easy to use. Now, when we started thinking about how we might design an entirely new platform, the concept of really depending upon a single molecule approach came to our forefront. And there are a couple of reasons why a single molecule approach is really powerful. The first is that it is definitionally the most sensitive a platform can be. A single molecule counter, literally measuring every single molecule. You cannot be more sensitive than a single molecule counter. Additionally, something really special happens at the single molecule level, which is that identification of molecules, figuring out what each molecule is the same as quantification. You just count up your identifications to get quantification. This is unlike other platforms where you might have to go through an intensity or a brightness in order to get quantification. We also recognize that a single molecule intact protein approach doesn't suffer from the challenges of peptide to protein assembly. And so protein measurement and quantification is both more straightforward and also potentially more accurate. Lastly, when thinking about things like proteoforms, looking at multiple modifications on an individual molecule, this simply isn't possible with a peptide-centric view, where you're taking all of those different modifications and splitting them up across peptides. So our goal with the Nautilus platform really was to build a single molecule approach that led to simple, interpretable, quantitative output, something like this, where we might end up with a protein identity, how much of that protein is present in a sample, as well as a statistical measure of confidence. The platform has three key components to it. The first is our single molecule library preparation and deposition approach. The second is an iterative approach for binding each of those molecules with a set of affinity reagents, imaging them, rinsing off those affinity reagents, and doing this cycle after cycle after cycle. And then the last is a data analysis and interpretation to take all of that amazing multi-cycle data, single molecule data, and infer protein identities and quantities. We're going to walk through each of the steps in this process over the next couple minutes. As I mentioned, the first part of the Nautilus approach is the sample preparation, the library preparation method that takes all of the molecules from the sample and deposits them one at a time onto a hyperdense patterned flow cell. The workflow is designed to be extremely simple and straightforward so that it's accessible to any biological researcher. And it's shown here on this slide. We start with a sample, which can be any number of different types of protein mixtures. We then denature that sample, which is really critical, using STS. We functionalize the lysines and primary amines using an NHS methyltetrazine group, which puts a click reagent onto each of the proteins. And then we further denature and reduce the disulfide bonds and protect them to help keep the proteins unfolded. All of this is done as a single pot reaction. We then do a few cleanup steps to remove the reactants. And at the end of that, we're left with a series of protein molecules from our sample that have been labeled with a click methyltetrazine group. We then mix them with our proprietary nanoparticle scaffolds and then the proteins will then attach. What's really special about this nanoparticle scaffold is it's been designed to have exactly one conjugation site. So on the proteins, you have a methyltetrazine. On the scaffolds, which look a little bit like Triscuits with a flagpole sticking out the top. At the top of that flagpole is the other half, the TCO. The methyltetrazine conjugates to the TCO in a covalent reaction. We then just, again, do a cleanup step and take all of those protein nanoparticle scaffold conjugates, flow them over the flow cell, and they deposit on a series of landing pads. We took this approach because it allowed us to generate a hyperdense protein array where we could be quite confident that we had single protein molecules immobilized at each landing pad in the flow cell. What's so exciting is being able to look at the flow cell itself. And so what we see in this picture on the left is an incredibly zoomed in view of our flow cell. Each one of these individual little blobbies is one molecule from the sample. And what was so critical to us and why we've put so much effort into this is that we recognized early on that measuring a tremendously large number of molecules was critical to get the dynamic range that we wanted to be able to match the scale of the proteome. On the right, what we're showing is a simulation where we examined how different, what the effective dynamic range of a sample might be on the platform. In this case, taking a model of what serum looks like as generated by David Fenio et al., and estimating how much of that sample could be deposited on the flow cell as if we had on the order of billions of molecules. And what's so special about the platform is that once a molecule is immobilized, it becomes its own universe and can potentially be identified and quantified. Once we have the molecules immobilized, the second step in the process is the measurement of each of those individual molecules. And so as we see here in this video, we immobilize the molecules. We then come in with fluorescently labeled affinity reagents, multi-affinity probes, that bind to in a plurality of proteins. Each one is fluorescently labeled. On the right is the primary data that you can see coming from the platform, these sort of digital starry skies. And we bind, take a picture, and then remove, and then introduce another reagent and go through that cycle over and over and over again. Each one of these probes is not protein specific. In fact, we've designed them to be explicitly recognizing very short epitopes, 3 to 4 amino acids long. So while any one probe is not particularly specific, the combination of binding of multiple probes to an individual molecule allows you to infer or decode the identity of a protein. So for instance, if we look at one particular molecule, we might have a pattern of binding where we had binding with probes that recognize the epitope KVK or NMS or YTH, etc. And we build this pattern. We can then ask a question, given what we've observed, given this pattern of binding and what we know about the probes and what epitopes they recognize, what protein is compatible with this pattern of binding, and assign it a probability. This is the heart of the PRISM method. Once we've identified each individual molecule, we can then take those identifications and count them up to get to our quantification. So the overall workflow is we perform our sample preparation, we then do our iterative multi-cycle imaging, generate these binding vectors through our machine learning process, infer the protein identities, and then add up all of those identities to get our quantification. In the next portion of today's seminar, I'll dive into a couple of the common questions we get. And so among the biggest questions we get are really about the multi-affinity probes, these probes that recognize short epitopes, 3 to 4 amino acids long. Typically, one of the biggest questions is, well, how many of these do you need? We recognize that they're on the order of 20,000 or so proteins in the proteome. And if we were to use traditional affinity reagents that are protein specific, we would need 20,000 or more different affinity reagents to measure all the proteins. But what's really unique and special about our method is that by using these short epitope probes, we're potentially able to discriminate and measure substantively all of the proteome using 200 to 300 of these probes. The intuition behind this is very similar to that of the game Guess Who. So in fact, you can think of this as a massively parallel machine learning based version of Guess Who. So for instance, if we had a protein at a spot, we were trying to figure out who it is, we could use very, very specific affinity reagents to figure out its identity. But it would take us a large number of affinity reagents. We'd have to ask a large number of questions to figure out what protein was present. On the other hand, if we ask a series of much more general questions, like, is it a mammal? Is it a large mammal? Or a cat? Or a woodland creature? We're able to get to the answer incredibly efficiently with a smaller number of questions. That's exactly how our PRISM method works. The next question we get asked is, well, how do you actually build these multi-affinity reagents that recognize short epitopes? And the answer is we spent several years building an incredible pipeline to do this. And so it starts with candidate discovery, which is typically a phage display or CLEX process, followed by validation of binding, testing on our instrument itself, and then additional characterization. One thing I'll really emphasize is just how deep the characterization of our affinity reagents is. So as I mentioned before, we start off with a phage display workflow that was built in-house over many years. We next validate that each of the affinity reagents binds to the target it was created to recognize. And we often get asked, well, do these bind strongly? Are these weak binders or strong binders? And what you can see from just 3 of our examples here that recognize either 4 amino acid or 3 amino acid epitopes, that they are incredibly strong binders, picomolar strength binders, EC50s. After we've validated that they bind to the target they were selected against, we then go and do additional work validating that once they're labeled, they continue to work. And then we do a tremendous amount of work to validate what they bind beyond the target that they were selected for. We know that affinity reagents are typically extremely cross-reactive, and that's totally fine. Our platform is very happy with that. But we want to know what that cross-reactivity is. So we'll challenge them with millions or even billions in some cases of different peptide targets and ask which are the peptide targets that it has the best affinities for. And so here are a couple examples. This was an affinity reagent that was raised against the epitope histidine, threonine, proline. And you can see it does recognize peptides containing histidine, threonine, proline. But it also recognizes the biophysically very similar histidine, serine, proline, and then to a lesser extent, histidine, valine, proline. And this extent of characterization is something that's very special about the reagents on our platform. Another question we often get is, well, we know you're denaturing the proteins during your sample preparation, but still, are these probes able to recognize buried regions in proteins? Are they able to recognize what used to be secondary structure regions in proteins? Do they have a bias between the N and C terminal portions of a protein? What I'm showing you here are two proteins, transferrin and G6PI. Highlighted are different regions of the proteins that we've shown binding to on our instrument. And you can see they're scattered uniformly throughout the protein, and they are also, there's no particular N to C terminal bias. The next place where we often get questions is about the stability of the instrument and the platform. So this is something that we've been working really hard over the last couple of years to optimize. But remember that within the instrument, there are a series of cycles of measurement that are performed. First, the proteins are immobilized in the flow cell. Then we introduce our fluorescently labeled affinity reagents, rinse off the unbound portion, and then take a picture. We then wash off all of those probes to clean the surface, and then we introduce a second affinity reagent. We go around this cycle over and over and over again. So some of the questions we get asked are really about those protein scaffold conjugates. Do they stay stuck on the surface? One can imagine that over many cycles, perhaps they fall off. And what you can see here, and please do notice the axes is really tight, that we've shown over 100 cycles that they really do stay very sturdily on the surface. And that's, again, something we've optimized. The next question you might ask is, well, are you potentially damaging the proteins in some way? And so that your ability to bind declines across cycles. And again, we see no evidence of that. We see that it is quite consistent across many cycles. The other question we get is, are you damaging the surface in some way or failing to remove affinity reagents? And so the background is it building up? And again, through tremendous optimization, we don't see that occurring. So we feel like we've substantially improved the stability of the platform. All right. I'm going to change gears now and dive into some of the advances that we've had on using the platform for broad scale studies. And in particular, I'm going to dive into one larger scale experiment in which we used either transferrin or G6PI, glucose 6-phosphate, some model proteins, no protein, or mixtures of them. And we deposited those into different flow cell lanes and performed our analysis. In this particular study, 36 distinct multi-affinity probes recognizing different epitopes were used, and we randomized the probe order. What you're looking at down below is our three different proteins in the experiment, and the darkness of the cell in this matrix is reflective of how strong the recognition is of a probe to that protein. So, for instance, of what would be expected based upon our prior understanding and our prior characterization of the affinity reagents. So, for instance, what we would anticipate is the probes that are being used in cycles 14 and 15 should have very strong binding to G6PI, whereas the probe in, say, cycle 13 or maybe cycle 22 is weaker. One of the other things I'll just point out is that these patterns are quite distinct between our different proteins. And this is to be expected because, of course, the sequences of the proteins are different, and therefore the epitope binding and recognition is different. So just to dig into what the actual raw data shown here on the right is that we image the flow cell. You're looking at just one small portion of an image. In total, this experiment was quite large, tens of thousands of images over the 36 cycles. We did validate that the proteins themselves stayed immobilized over the course of the experiment, and the background binding rate stayed very low. But even on a prototype flow cell and a smaller number of cycles, 36, 40 cycles, this is a tremendous amount of raw image data that's collected. Once this data has been collected, it then goes through a series of image processing steps, denoising, deskewing, alignment, that then allows us to take this data and boil it down to binary digital data, where in each, what you're looking at here is the trace for one molecule on the surface. And you can see that there are a wide variety of different patterns that molecules can have. And this is because it is a single molecule, highly stochastic system. And the machine learning approach under the hood is designed to take advantage of that. Once we've boiled these down to these binding vectors, we then go forwards to identify each protein. One of the things that's fun to look at, if we take just the top 5 most prevalent binding patterns for each of our 2 proteins here, transferrin and G6Pi, you can notice that there are positions, different probes. For instance, in cycle 14, this is a probe that equally well recognizes both transferrin and G6Pi. So it's not hugely differentiating. On the other hand, you have other probes like the one used in cycle 17 that is dominantly associated with binding to transferrin and almost never seen in G6Pi. This is the raw substrate, which we use to figure out the identity of a protein. One of the ways that we build confidence in protein identification is by looking at, sort of going inside the machine learning itself and saying, all right, well, how confident are we that this particular spot, this particular molecule is transferrin versus G6Pi? And so what I'm showing you here are 2 different traces of 2 different molecules. The top is transferrin, the bottom is G6Pi. In triangles are noted where there were different binding events. And the line is tracing our confidence that the protein at this particular location is either transferrin or G6Pi. So what you can see is that there are particular places like here that are shown in blue. These are places where we would anticipate our binding is more likely to be indicative of transferrin than G6Pi. We get a boost in confidence when we get binding events at those locations. On the other hand, some places where we get a binding event, like here in cycle 14, this is a location that's equally likely to be bound, a probe that's equally likely to be bound by either transferrin or G6Pi. So we don't see a boost in our probability here. But over the course of many cycles, we end up multiple log more confident in the identity of this molecule being transferrin versus G6Pi. We can accordingly look at a different molecule that was assigned to G6Pi, and we see a different pattern of binding, again, lighting up in the more orange regions, and again, seeing substantial log differences in binding patterns. One thing I'll just point out about this approach is when we think about data quality, we really think about how much information is used in making identifications and quantifications. And so on a Western, of course, there's typically one antibody that's used in ELISA, typically 2. By mass spectrometry, we get a range of different pieces of information for any given protein, the majority of which being identified by a single peptide, and then sort of an exponential decay from there, with the most abundant proteins being identified with a smaller number of touches from there. On our platform, if we look at the amount of information that's used to identify any given molecule, the vast majority of proteins are getting identified with dozens of different touches, which gives us additional confidence in an identification. One of the other ways that we assess the confidence of an identification is by diving in and asking the question, well, what would happen? We had an observed binding pattern. What would happen if one of those binding events, for instance, were a non-binding event? Would we still be confident that the protein that was assigned was correct? And in this case, if we were to take out this one identification or this one binding event and turn it to a non-binding event, the most prevalent protein to come back, the most confident protein, would still be model protein 1. We can also take non-binding events and turn them to binding events, such as here and here, or change many more. But we can explore this in silico to build, again, our confidence in an identification. And so what I'm showing you here is, again, for one particular molecule, how we build confidence in that identification. This molecule, before there was any appreciable percentage of identifications that would have been flipped, it would have taken 6 binding events to have been flipped to non-binding events, or 4 non-binding events to be flipped. So we would call this an incredibly confident identification that's very robust to the pattern of binding and even variations in what was observed. Not all identifications are this confident. We have some, for instance, this one, where even one change in either direction, flipping a non-binding event or a binding event, leads to a substantial percentage of binding events flipping. One of the other areas that we've really dug into is assessing the false discovery rate. I'm not going to spend a ton of time here, except to say that we are taking a page from the book of mass spectrometry using a target decoy-based method. In this approach, we generate decoys that are meant to be reflective of real proteins. We've tried a number of different approaches, ranging from shuffling of proteins to alternative proteomes, as well as paired decoys. And we evaluated them using simulated data. They all work reasonably well, though shuffled proteins and alternative proteomes seem to work the best and were resilient to the most expected failure modes of our platform. Once we have a confident set of identifications, the next step is taking those identifications and counting them up, as I mentioned, to determine the quantities of each protein in our sample. And so to examine quantification on our platform, we first took 2 samples with a mixture of proteins, and I'm just showing you 2 of the proteins here, where within one sample we had a given amount of transferrin and another protein, PKM2. In a second sample, we changed the abundances of those. And we can look at the changed abundance from sample 1 to sample 2, and that was expected from the sample composition. And if we look at transferrin, there was approximately a 6-fold difference in abundance of transferrin between sample 1 and sample 2. PKM2 had about a 2-fold difference in abundance between sample 1 and sample 2. And you can see the quantifications are pretty close there. One of the other things that's very exciting about the platform is the ability to look at within a sample. And so within sample 2, we can look at the ratio of abundance between transferrin and PKM2 and see that there's about a 2-fold difference, and that was also captured. One aspect of the platform that's great is that one can also turn a knob, and for every given spot, every given molecule, we have a probability estimate of the identification. And so one can tighten those probability thresholds or FDR thresholds in order to only look at the most confident identifications. And that can allow you to remove false positives and drive down sensitivity, or and drive down quantitative accuracy to low amounts, or to ensure that you have the widest net to capture as many proteins. Well, the last question we really wanted to look at was, as a single molecule platform, we would anticipate that the platform could be extremely sensitive. And so we did a simple titration series and looked at either a protein in a background of empty nanoparticles or in a background of another protein, and examined the limit of quantification of the platform. We're really excited to see that that limit of quantification on the flow cell was in the yoctomolar range. And so tremendously exciting sensitivity. We also examined the reproducibility of the platform, running the platform many times across multiple days and flow cells, and found a CV of less than 10%. One other question that comes up is, well, could you have gotten this sensitivity? Did you need all of this infrastructure? Did you need this multi-cycle? Could you have just gotten the sensitivity with one antibody? And the answer is very clearly no. So what I'm showing you here on the right here is if you had used just a single one of our multi-affinity probes and attempted to do quantification from that, your limited detection is substantially worse. And this is because of the leverage of the power of being able to really make confident distinctions between false positive detections and false negative detections. As one increases the number of probes, your confidence can grow even more and more and more. One last point I'll make about quantification, coming back to what I mentioned in the beginning, is that the larger the flow cell, the more molecules that are measured, the wider the dynamic range of the platform. Typically, when people think about dynamic range, they think about it as the difference between the least abundant species and the most abundant species, and that's what's shown here in orange. And you can see that this grows until saturation as one increases the number of molecules that are measured. We often think about dynamic range less in terms of those extrema, but instead as the point which you're measuring 90% or more of the molecules that are present in a given concentration band, and which we've shown here in gray. And you can see that this tracks roughly linearly with the number of molecules that are being measured. So I'll just summarize quickly. Really, what I hope I've shown you today is that we've built a robust platform for characterizing multi-affinity probes that bind to a variety of different proteins within our instrument platform. We've been scaling up the stability of the platform for iterative interrogation of individual molecules over and over and over again, as well as focusing on robustness of our false discovery rate methodology so that we can be really confident in the results that come off the platform. Shown reproducible quantification with extremely high yoctomole sensitivity. And in the future, we're looking forward to continuing to improve both the number of molecules that we measure, the cycles that are measured, the stability, sensitivity, and breadth of proteome measured. With that, I'm going to pause and hand it over to Andreas to dive into the tremendous advances on the proteoform side.
Parag has just demonstrated how we use the Nautilus proteome analysis platform for broad-scale decoding of proteomes. I will describe how we use the same platform in a slightly modified fashion to do proteoform analysis. Like other proteomics methods, our platform has two modes of operation, with a discovery mode and a targeted mode. The difference is simply what types of affinity reagents we use to probe the sample. For the discovery mode, we use a novel of multi-affinity probes that Parag described here on the left. For the targeted application, we utilize the same platform, but switch to site-specific and target-specific probes that are commercially available. So by simply reusing millions of available antibodies, we can extend the functionality of the platform to study proteoforms of important molecular targets. So before I describe how we utilize off-the-shelf reagents validated for proteoform analysis, let me describe first what proteoforms are and why they are important. Proteoforms originate from a combination of protein splice isoforms and post-translation modifications. There are potentially millions of proteoforms in the human proteome as a result of about 230 different post-translation modifications on top of 70,000 splice isoforms from the approximately 20,000 genes in the human genome. We know that post-translation modifications affect the structure, function, and distribution of proteins, which means the abundance of proteoforms and their specific patterns of PTMs are biologically relevant. The question you might ask, if proteoforms are so important, why do we not measure them routinely? The answer might be easy to understand if we compare the protein molecules in the next slide on the left panel with the molecules in the right panel. As you can probably see, the 3 protein molecules on the left are different from the ones on the right. However, if you use traditional proteomics technologies, particular peptide-centric approaches, on average, you would be unable to differentiate the left from the right. Even for top-down mass spectrometry methods, it is very challenging to determine the differences in proteoforms. What is also apparent in the comparison above here is that proteoforms on the left are biologically distinct from the right. Those may have discrete biological functions, but it is analytically hard to measure. So today we have a lack of understanding of their functions because of our lack of tools to differentiate proteoforms analytically. So how might we be able to change that? The Nautilus approach to proteoform analysis utilizes the flexibility of the single molecule platform and the ability to utilize iterative cycles of binding and releasing of high affinity reagents to establish the presence and the quantity of proteoforms in the sample. So by utilizing the multi-cyclic capabilities of the platform to probe each molecule in the flow cell multiple times over hundreds of millions of individual molecules, we create a binding pattern for each specific high affinity reagent through recording the binding events of 4 cycles in this case. By applying a correction method to account for cross-reactivity, we are able to determine the specific proteoform and the quantity in the sample. So let me walk you through an example how this works. As an example of proteoform analysis, we apply our approach to the tau protein, a protein critical in development of many neurodegenerative conditions. The tau protein lends itself well to this type of analysis because it is known to have isoforms and many PTMs. I should also add here that this is the focus of our collaboration with Genentech. So we select a number of specific key sites in the tau protein known to drive biology, but where the underlying contribution of proteoforms is unknown. To do that, we create the recombinant forms of 2 of the tau isoforms, the 0N and 2N. And based on those 2 isoforms, we also create a specific subset of PTM-modified proteoforms by phosphorylating the 2N tau protein in a treatment with protein kinase A, and proteoforms of 0N by phosphorylation with ERK. Then we characterize those standards by LCMS to validate site specificity for the phosphorylation and their relative abundance. With the detection of antibodies for tau proteoforms in hand and the respective control standards, here's the experiment we run to detect and quantify tau proteoform standards. So we place the following sequence isoforms and modified isoforms in lanes 1 to 4 on the flow cell, as shown on the left. Then we run the experiment by detecting binding patterns for millions of single molecules on the flow cell with a panel of 4 detection antibodies that are specific to those proteoforms in the control sample. Then by analyzing the binding pattern from millions of single molecule measurements, we're able to identify and quantify the proteoforms. After detection of the signal, the next critical step in the analysis is the correction of the data for antibody cross-reactivity. What is antibody cross-reactivity, you may ask, and how do we account for it? It's well described that affinity reagents in general and antibodies in particular exhibit cross-reactivity. So there are 2 types of measurement errors that we have to account for due to that cross-reactivity. There's false positive errors, which occur when a binding signal is detected, even when the targeted epitope is not present. For example, the antibody binds to the incorrect epitope or non-specifically to parts of the flow cell. A false negative error occurs when a binding signal is not detected, even though the target epitope is present. What we observe on our platform is most errors are false negative errors rather than false positive errors. So we can take the approach to learn about the binding errors from a control standard, creating a binding model, and then applying that model to our sample lane to correct for those errors. Sounds complicated, but let me show how it works in detail. On the left panel, you see the expected signal from the Nautilus Proteome platform when we measure lane 3, which contains the proteome standard 2 and 4R phosphorylated at S214. The middle panel shows what we actually measure in 4 cycles of measurements with our antibody panel, which includes false positive and false negative binding errors. Then finally, on the right panel, you see the results of the proteoform abundance for the 2 and 4R S214 sample in lane 3 after the application of the binding model correction. As you can clearly see, we can recover the true proteoform abundance that is known to be present in lane 3 through the statistical correction. Now that we have demonstrated that we can correct for errors by learning from control standards, can we apply this approach to a mixture of proteoforms? To answer this question, let me show you in the next slide how we did the experiment. So we repeat the experiment I just described above, but this time we add the mixture of tau proteoform standards on lane 5 at a molar ratio shown on the table to the right. We also expand the antibody detection panel with a 5th antibody that's specifically probing for phosphorylation site S on serine 396 on the 0N isoform. To better understand the results, let me walk you through the graph above. The lower panel shows 32 potential proteoforms that you expect to detect using a 5 antibody detection panel. Calculation simple, to the power of 5 is 32. The specific epitopes that we detect are shown on the y-axis of the lower panel and are indicated by the connected dots. The upper panel shows the relative abundance of the proteoforms in parts per million. Starting out with the 0N proteoform in green present at around 100,000 ppm, compare that signal with the abundance of the 2N proteoform at around 60,000 ppm in purple. You notice they're approximately half of the abundance of 0N, reflecting the molar ratio for those 2 isoforms expected in the mixture. Now comparing the purple trace to the 2N S214 abundance highlighted in blue, which have the same molar ratio than the 2N form in purple. We see that the abundance are comparable between those 2 proteoforms. Now for the most abundant proteoform at a molar ratio of half of the mixture, the abundance of about 200,000 ppm is about twice that of 2N. However, what you also notice is that the 0N proteoform modified by ERK expect to be modified at position 181 and 396, and therefore has several related proteoforms in the mixture. And we can also detect that as anticipated. However, you can also see there is 2 more additional isoforms that we can detect, indicating that there's a phosphorylation event at 214. That was unexpected. However, when we checked the LCMS mass spectrometry results for the standards, we found that the 0N ERK modification also phosphorylated that some of the molecules at site 214, which is clearly indicated here in the proteoform mixture analysis. This is an overall good match between the expected and measured abundance for tau proteoforms in the mixture. So now moving from mixtures of standards to biological samples, can we detect tau proteoforms in cells endogenously expressing tau proteins? For that, we actually looked at HEK cells that had overexpressed particular proteoforms of tau. On the panel to the left, we look at results of specific tau proteoform detection in a HEK cell lysate overexpressing 0 and 4R tau and 2 and 4R tau on the right. On the left panel, the presence of the 0N proteoform was detected with the anti-0N antibody with limited off-target detection as determined by the anti-2N antibody. And similarly, the 2 and 4R tau overexpression was confirmed with the anti-2N antibody, and limited off-target detection in that cell line was determined with the anti-0N antibody. So this results clearly demonstrates the specificity of proteoform detection of endogenous tau proteins in a biological model system. Maybe in summary, I can confirm that we demonstrate the detection of distinct tau proteoforms at a single molecule level. I also showed you that we can overcome errors associated with antibody cross-reactivity by constructing a binding model that corrects for those errors and obtain true proteoform abundance. By exploiting the massively parallel nature of our single molecule platform, we correctly determine the relative abundance of tau proteoforms in the mixture. So moving forward, we can define the proteoform heterogeneity that underpins complex biological samples using our massive parallel capabilities for single molecule measurements. So one of our next steps obviously includes the application of this method to human cell lines, brain or CSF tissues from patients' samples with Alzheimer's disease. Looking back at the agenda of this presentation, I hope that we have demonstrated that the Nautilus proteome analysis platform aims to create a new level of depth in proteomics analysis across a number of application areas that we have highlighted here. But we also hope that the platform with its unique capabilities can unlock novel insights for your research. So I encourage you to reach out and let us know about your specific research questions. And as you think about your research question, keep the design criteria for the Nautilus proteome analysis platform in mind. We aim to provide a sensitive proteome analysis platform with a wide dynamic range and the ability to quantify the entire proteome based on billions of single intact protein measurements per run. Of course, the digital nature of the measurements provide reproducible and robust quantification at either proteome-wide level or for individual proteoforms, as we have shown. With our ability to deliver integrated and easy-to-use workflows for many samples from your study with a runtime of a typical working day, we hope to unlock proteome measurements in many laboratories that don't have access to proteome analysis today. And with that, I thank you for your time, and let's turn to some questions.
Thank you, Parag and Andreas, for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located in the lower left corner of your screen. We'll answer as many of your questions as we have time for. Let's go ahead and get started. Our first question, it says, I appreciated the results that you have demonstrated for the tau proteoforms. Can I analyze proteoforms of the protein that I am working on?
Yes, I can take that. Thanks for the question. So we can detect proteoforms for additional proteins of interest as long as there are antibodies available that are associated with the specific epitope of interest. Currently, we're developing methods to be able to incorporate the additional content that customers like yourself may be asking for. And so we use millions of available antibodies that we validate for the purpose.
Great, thank you. Our next question here says, great presentation, quick question. For proteoform detection, am I understanding it correctly that you're sequencing the antibodies that bind to tau and not tau itself?
We don't sequence anything. We look at the binding patterns of these tau-specific antibodies in the example we have shown, to the actual tau protein that's in the flow cell. So there's no amino acid type sequencing ongoing in this experiment. It's just a binding event observation.
Excellent, thank you. Our next question that we have here asks, how does the platform deal with post-translational modifications or single amino acid variants in broad scale mode?
It's a great question. So there are a couple different ways that we anticipate being able to support post-translational modifications and amino acid variants. The first on the PTM side is that, as Andreas mentioned, we can import a variety of different antibodies that already exist. So for instance, there's already a great or several great anti-phosphotyrosine antibodies that are available. And so those would be readily importable onto the platform. For amino acid variants and other types of PTMs, there is information in both the binding and lack of binding of an affinity reagent. So what you can think of is you need a certain number of touches of the multi-affinity probes in order to identify the protein at all. And then any additional touches beyond that are really instructive in helping you map the finer detailed aspects of the protein, including where you might have expected to see binding and you didn't. And that perhaps gives you information that there might be a PTM or a single amino acid variant. Of course, you can also develop specific probes that target those variants. So for instance, in KRAS, you may want to specifically include a G12D, G12C targeting affinity reagent so you can get that finer detail.
Excellent. Thank you. All right. Our next question here asks, how well does the current platform work for complex proteomes, i.e. cell lysates?
Yeah, so where we are in our platform development is we've been ratcheting up the diversity of model systems and samples that we've been working with, increasing the stability of the platform to larger number of cycles that are required to decode proteins from more complex materials, more complex matrices samples, and are working our way through increasing sets of complex samples, starting with cell lysates and then ultimately going beyond.
Wonderful. All right, our next question here says, can this instrument detect very low levels of a specific protein in high protein matrix, such as plasma?
Yeah, great question. So the data that we showed really was speaking to the limit of detection of the platform and going to extreme sensitivity in the yoctomolar on chip range. That I would consider to be extremely high sensitivity. Then the question is, how does that relate to dynamic range in a high protein matrix? One of the things that's very special about a single molecule platform is that each molecule is its own universe. And so really the dynamic range is directly related to the number of molecules deposited on the flow cell. And so the later slides really were speaking to linearity in response between the number of molecules measured, intact protein molecules measured, and the effective dynamic range. So even for something like plasma, your ability to be ultra sensitive is really a function of that yoctomolar range of what can you actually immobilize on the platform on inside the flow cell.
Wonderful, thank you. Okay, our next question here says, how long does it take you to generate the multi-affinity probes, and is it cost effective to generate new probes required for certain applications?
So I think the best way to think about that is that we've spent years building a very robust process for not just building these affinity probes, but also for characterizing them. While at this time we aren't planning on broadly making new affinity reagents for specific applications, that is something that we do as part of our partnerships. And so as well as things that we're constantly doing in-house to expand the capabilities of the platform. So I'd encourage you to reach out to us directly to chat about possible partnering opportunities, because we're constantly excited by new questions and challenges, and have built a really remarkable team that is doing an amazing job both building new affinity reagents and characterizing them extremely deeply.
Great. And we have time for just one more question, so I'll go ahead and ask that now. This question says, protein identification is based on expected binding, so a specific database is needed for each species proteome. Are databases other than human available, and how do you deal with an unexpected protein contaminant, such as integrated viral capsid protein or partial deleted sequence? And let me know if you need me to read that again.
I think I got it. And so the first part of the answer to that question is that is just to remind you that we raise the affinity reagents against short linear epitopes. So again, 3 to 4 amino acids long. So we do require a reference database, such as a FASTA file similar to what's done in mass spectrometry-based proteomics. But we then have been working on systems to try to, based upon the expected epitope, map whether that epitope is present in a protein, and then build a prediction about the binding based upon the epitope presence or lack of presence in a protein. And so in that case, so at the moment we're focused on human. However, there's every expectation that this should work on any organism for which we have a protein FASTA file database available. And then with regards to partial deleted sequences or proteins that exist in the sample but aren't in the database, one of the things we were really careful about in the machine learning approach that was taken is that we wanted one that would fail gracefully, so that if there was a novel protein in the sample that wasn't in the database, instead of guessing very confidently wrong, it would just fail and say, I'm sorry, I don't know what this is. And part of how you can think about that is that as you start getting more and more touches on proteins, it becomes vanishingly improbable for you to be an incorrect protein, because you just have so much information by which to infer its identity. And so we generally believe that we're fairly tolerant to cases where the protein's not present in the database. With regards to partial or deleted sequence, that comes back to my prior question about what happens for PTMs and others. There's a certain amount of information that's required to figure out what the protein is. But then if there are alterations such as splice variants or deletions, those would show up as like, oh, hey, I would have expected this to bind, but it didn't. And there's potentially information there to help you get that finer grained mapping.
Okay. Thank you, Parag and Andreas, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Nautilus Biotechnology, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by our speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.